Well, welcome everybody to ICMDA webinars this afternoon. I'm Dr. Peter Saunders, the Chief Executive of the International Christian Medical and Dental Association. And today's webinar is on the very topical subject of COVID vaccines, effectiveness, safety, ethics and deployment. And we're privileged today to have two very eminent professors in this area speaking to us. First of all, Professor Anneliese Wilder-Smith, and secondly, Professor Gagandeep Kang. And I'll introduce each of them as we go along. So without further ado, let me introduce Professor Anneliese Wilder-Smith, who is the Professor of Emerging Infectious Diseases at the London School of Hygiene and Tropical Medicine. She also serves currently as consultant to the World Health Organization, Geneva in Switzerland, where she's the focal point for SAGE deliberations on COVID-19 vaccines. Anneliese specializes in emerging viral diseases such as Zika, dengue, chikungunya, yellow fever, SARS, influenza, and now, of course, COVID. And in 2003, she was at the forefront of the SARS outbreak in Singapore. With a career spanning almost 3 decades, she has led and co-led various clinical trials, published more than 350 scientific papers, edited and co-edited textbooks and travel magazine books, served on various editorial boards and scientific committees, including being editorial consultant to The Lancet. So let me hand over to you, Professor Wilder-Smith. We're very grateful to hear you. Thank you.
So thank you for the invitation, Peter. I'm really delighted to talk to you today. In fact, I feel very honored to be invited. Our family lived in Asia for more than 20 years, and I often visited CMC Vellore during that time. Therefore, I'm really absolutely thrilled to share today's session with Professor Gagandeep Kang from CMC Vellore, also known as Cherry. And Cherry and myself, we meet quite often. We both serve on the WHO SAGE working group on COVID-19 vaccines. And in fact, I will now focus my talk on some of the WHO deliberations around COVID-19 vaccines. So I'm really presenting some of the WHO processes. So as of 11 December, the cumulative number of COVID-19 cases notified WHO was close to 70 million, with sadly more than 1.5 million deaths. Just in 24 hours, there were more than 600,000 confirmed cases and 11,000 deaths. For corona skeptics, excess mortality has now been reported in basically all countries that have not successfully contained their national outbreaks. And in the US, in terms of absolute annual numbers, COVID is number 3 cause of death. So first cardiovascular, then cancer and COVID. But as of last week, COVID is now number 1 cause of death per day. So it has overtaken cardiovascular disease. You can also see on the side on the right that the epidemic curve is strikingly different by country and by region, underpinning again how this disease is driven in clusters by population densities and urbanizations, but mostly the epidemic curve really reflects the extent of the pandemic response that different countries have implemented. We also need to keep in mind some countries have successfully contained the outbreak, and these countries include China, New Zealand, Australia, Singapore, Taiwan, Vietnam, South Korea, and many others. While numbers of cases may not always be so accurate because it depends on testing capabilities, I think the number of deaths per 1 million probably give us a better or more accurate picture. And really it's the US, Brazil, Iran, and basically most of the European countries that have the highest deaths per 1 million population. Lockdowns are successful and have been successful. This is a disease that can be mitigated. but obviously they're not a durable solution, and all populations are still at risk, given that we have by far not reached herd immunity. We also know countries cannot beat COVID alone, as we are so interconnected, and what happens in one country will also affect the others. So there is therefore a real strong case to act. And the access to COVID-19 tools is called Accelerator, is a groundbreaking global collaboration to accelerate the development, production, and equitable access to vaccines, to therapeutics, and to diagnostics. And as you remember, COVID was declared a public health emergency of international concern on the 31st of January, and by 24th of April, ACT was already launched, and by now more than 186 countries have signed up to it. And it's a very, very important initiative. So COVAX is the vaccines pillar of the ACT Accelerator. And it really, and most countries in the world have joined. About 86% of all the countries, except for the United States and Russia. China also did join. And these are the goals of COVAX: to end the acute phase of the pandemic by the end of 2021, to deliver 2 billion doses by the end of 2021, to guarantee fair and equitable access, and also to support the largest portfolio of vaccine candidates that we've ever had globally. So the COVAX facility allows countries secure access to vaccines. So at its core, it actually is a risk-sharing mechanism. So we have a whole series of platforms. Some vaccines may be successful, some not. So we are all investing into this platform where some may fail, some may win, if that is the right word. And so we then all have access to these vaccines. And by participating, any country can participate, and participating countries will then receive access to those vaccines procured through COVAX at a pre-negotiated price. And we also, COVAX allows a lower to middle income countries, and they will then be supported through overseas financing agencies. So there are three components that inform countries to formulate the in-country vaccination strategy. So at WHO, we have, or at part of the COVAX facility, we have an allocation framework that has for months already thought through how to set the frame and the priorities for vaccination. Then, of course, we need the whole regulatory approval process that particularly looks at efficacy and safety. And then Cherry and myself, we work, we are members and the focal point for the SAGE, that's an advisory group of experts to help guide, develop policy formulations. But obviously, in the end, it's the countries themselves that are responsible for the final decision how to allocate and how to formulate their vaccine strategies. So this is what the allocation framework at WHO has put up. So the overall goal, obviously, is to protect public health and minimize the societal and economic impact by primarily initially focusing on COVID-19 mortality. We know there are lots of uncertainties, but one thing is certain. We certainly will have limited vaccine supply initially. So given the extremely limited vaccine supply, the priority is to initially focus on health and social workers, with 3% given with vaccines given to 3% of the population size of every country, followed then by covering high-risk adults. So the idea is that every country through COVAX that has signed up to COVAX would receive doses proportional to that total population up to 20%. And this is, and the aim is to get those 20% latest by the end of 2021. Obviously, then there's good potential that we may have more vaccines, but again, then it would, then the allocation would depend on the epidemiological situation in countries and on the vulnerabilities of countries. So obviously, the COVID-19 crisis required an accelerated pathway. Whilst we all know that in the past we worked sequentially, and often there were gaps between phases, and which then led to what we often now refer to as a 10-year pathway, we needed to compress the timelines. But it's very important to repeatedly state that compressing timelines does not mean cutting corners. It means we are working faster. We try not to be sequential, but work in parallel so that we prepare steps in parallel. And very important is also why were we so successful for this COVID outbreak? And I think there's several points we need to state here. We already had CEPI in place. CEPI is this Coalition for Epidemic Response, and so we already had it in place after H1N1. They had already worked on platforms. There already had been a governance structure, a financing structure, and we all worked around the clock. And I think important, whilst we have compressed the timelines, we did not cut corners on the phase 1, 2, and 3 trials, and it's important. And remember, the duration of a trial depends on the sample size of your trial, but also the attack rate. And the attack rate for COVID-19 is pretty high compared to many other diseases. Therefore, to accrue a certain number of events was done much faster than we've seen for other vaccines. It's also important from us, from WHO, for SAGE. Normally, we as a SAGE working group only start working after vaccine has been licensed. In this case, as Cherry can witness, we started working basically already, I think since June, to already have all the processes in place so that once there is a license or even only emergency use authorization, that we would be immediate with our policy recommendation and not take another year to think about it. Let's move to the vaccine landscape. So obviously we need all platforms, and some will succeed. Every platform will have its own advantages and disadvantages. But currently all platforms are being used, well-established ones as well as novel ones. So the well-established include the virus vaccines, live attenuated or inactivated, protein-based vaccines, protein subunits or VLP, and now the more novel viral vector vaccines, which includes replicating and non-replicating vaccines, and the really novel ones, nucleic acid vaccines, which include DNA and RNA vaccine. Let me just delve into the two newer ones, the viral vector and the nucleic and the mRNA vaccines. So for the mRNA vaccine, both these platforms, they just select some bits, some parts of the genetic material that signals how to make the spike protein. We now know that the spike protein is the immunogenic antigen that we need to target. So only the spike protein is included here. The rest of the code is not included, and therefore we think that these vaccines are actually safe or safer because they can't lead to the cell reproducing the whole virus. Now, in the mRNA vaccine, the blueprint of the spike protein is in the messenger RNA. And this is the molecule that delivers then the spike protein into the cells. For the Oxford vaccine, which uses an adenovirus vector, they also just use a spike protein into the genetic formulation of a carrier, and the carrier is then to human, a harmless adenovirus vector, and it's a chimpanzee vector. So now let's look at the overview of the platforms. And each has their own attributes or advantages and disadvantages. So for example, the RNA vaccine is just very fast to respond, to build up. And that's why the RNA was the very first to enter human trials, because you have an existing platform and you just replace the genetic code. Here it was a spike protein. But it's not a licensed platform yet. So we don't have regulatory experience with an unlicensed platform, with a hitherto unlicensed platform. So although the initial speed is fast though, we also don't have a very speedy manufacturing capability. Now for the replicating, for the viral vector vaccines, they're also relatively fast, and we probably have already a better manufacturing capabilities. Inactivated and live attenuated vaccines, by definition, need a longer time to prepare, because you have to find the exact dose, you have to do a lot of safety studies before you even can go into humans. But it's a licensed platform, we have experience with it, and we have manufacturing capability. So now these are the advantages. Here is the landscape of I think it's now more than 50 candidates in human clinical trials. And now quite more than 11 have entered phase 3 or have already, have already had their first efficacy readouts. Those in purple had their efficacy readouts. So every trial has a priori a pre-specified trigger of events. If you have reached a certain amount of trigger, you don't unblind, but an independent committee looks at the data. And this was published, as you know, in press releases to the world. That was AstraZeneca, it was the Russian Gamaleya, it was the Pfizer BioNTech, Moderna, and just recently also Sinopharm from China. So luckily, for all the COVAX candidates that we've seen so far that have entered phase 3 trials, all the preclinical and early clinical data looks so promising. Basically, binding and neutralizing antibodies are induced with most of these candidates. Also, very importantly, cellular immune response has been induced. Vaccine safety profiles look as if they're very generally well tolerated. And so far, most of these studies have supported the expansion to the large phase 3 trials. But there are also uncertainties. And so will the vaccine prevent just severe disease, or will a vaccine prevent infection and transmission, which is important for indirect protection, herd immunity, or will the vaccine cover both? Most of the trials do not address, and I state that, do not address the question on transmission. They just look at the primary endpoints or secondary endpoints on preventing clinical symptomatic disease and severe disease and deaths. What's the safety? Will the vaccine have differential efficacy? That means, will it be lower in older age groups? And we know that most vaccines have a lower vaccine efficacy in older age groups, and often also in those with comorbidities. How does a country react if you have several platforms and you have access to several vaccines, and you have to roll them out? And obviously they come at different stages. These are incredible logistic challenges. And we all don't know in the beginning how much vaccine we actually will have. We also don't know yet. Neither do we know the duration of the natural immunity, and of course not the duration of a vaccine immunity, as we only have limited observation time. So this is what we did at SAGE. We started, as I said, several months ago. That means we did not have access to vaccine-specific data. We were still living in these uncertainties. So we started very logically and very systematically to first address the values framework. What are the ethical and worldview basis? What is the basis for prioritization in a context where we will have initially limited vaccine supply, so that not every country, or if even with country, we all go berserk and have different vaccine strategies. Then we use the values framework to guide and develop what we now call the WHO prioritization roadmap, which target populations then to prioritize. And only when we have a vaccine, and we now do have a vaccine that is under some kind of approval, only then we will develop policy recommendations. So SAGE has not yet developed policy recommendations. We are waiting for the EMA, Emergency Use Authorization, which will take place end of December. So this is the values framework, the core principles. Obviously, human well-being is foremost. Not only do we want to protect and promote human well-being that includes health, but it's also about social and economic security, human rights, civil liberties, and very importantly, we also stress the importance of child development. Equal respect. We recognize and treat all human beings as having equal moral status and their interests as deserving of equal moral consideration. Global and national equity obviously refers to between and within countries to ensure equitable distribution. Reciprocity is the principle where we honor the obligations of reciprocity to those individuals and groups within countries who bear significant additional risks and burden of COVID-19 for the benefit of the society. And that's obviously here mainly the healthcare workers, but also essential workers who could not socially distance during lockdowns. And we also stress the importance of legitimacy, which make global decisions about vaccine allocation and national decisions about vaccine prioritization should be done through transparent processes. And these processes, we should have shared values, best available scientific evidence, and also legitimacy needs an appropriate representation and input by affected parties. What we do not want to see is certain groups or more privileged to have priority. Well, this is all well said and difficult to implement. So based on these principles we've then formulated 11 objectives. I won't go into these in great detail, but based on these objectives, we then worked towards thinking through the prioritization. Now we also need the science, obviously. What is the science for the prioritization? So here you see that if you look at who is at risk for most severe disease, it is, as we know, very age-driven. Although the bulk of disease in the left figure is clearly in the age groups of 20 to 50, the bulk of deaths increases with age. And in fact, it's also higher, far higher in males than in females. On the right, you see the infection fatality ratio, which clearly shows how suddenly it increases above the age of about 60. So I really warn against using generic infection fatality ratios. They should always be age-based infection fatality ratios. So clearly age is a big risk factor, but the age distribution differs from country to country. So here's a study that looks at the age distribution of COVID deaths in the US versus Andhra Pradesh in India. Green is India, purple is the United States. So you will see here, so the life expectancy in the US is obviously higher. It's about 79 years, and in India, it's about 69. The mean age is much lower in India compared to the US. So although the infection fatality ratio on biologically grounds is the similar, you can also see it here in E, is similar in any, or should be similar in any country, you will see that the bulk of deaths here in purple in the US is above the age of 75. But the bulk of deaths in India is in the age group of 50 to 64 or 65 to 74. So we have to take the population age structure into account. ESARIC is a network of clinical care providers in hospitals, and these are the data from 200,000 hospitalized cases, important, in the UK. And in these 200,000, 26% died. So once you have your hospitalization as your denominator, you know, this disease really does not look very good. So here they published a multivariable Cox proportional hazard model where hazard is death. And again, you see here that the hazard is highest based on age. So this is the strongest independent risk factor. Here you see female is protected or male has a higher risk. And then all these chronic comorbidities also increase the risk, but nothing increases the risk more than age itself. But obviously comorbidities and age are also confounding, they're correlated. So to get a feel for the number of vaccines you need to cover the population at highest risk, you need to understand the population structure. So Lancet published the global, regional, and national estimates of populations at increased risk. So every single country can actually look at this as this Lancet article and look what's the proportion of people above the age of 65. And on average, actually, this is about 20%. It's maybe 28% or higher in Europe, and maybe a little bit lower in lower to middle income countries, but this is an estimate. And if you also take comorbidities into account, even below the age of 65, then this number that needs to be vaccinated increases. But in addition to what we now know as risk factors, which is age and comorbidities, we know that exposure is also a risk factor. And sadly, initially, as we all know, PPE was insufficient, or we were not trained enough how best to use PPE. But according to Amnesty, 7,000 healthcare workers have died. And that it's really, I think it's a tragedy that should be avoided. So for over 7,000 people to die while trying to save others, it's a crisis on a staggering scale. So based on the science and our observation, there are 3 priority populations for vaccination: those at higher risk of severe disease, those at higher risk of exposure, and therefore also higher risk of severe disease, and those that contribute most to transmission. Obviously, there's a lot of overlapping. But again, if you look at these populations and to get a feel how much vaccine we actually need to cover those populations, here are the target groups by population size. The world's population is 7.7 billion. Almost 2 billion are children. They are excluded from vaccination initially, although they contribute somehow to transmission because they have a very low risk of severe disease outcomes. But if you look at the global population size above the age of 65, it's 700 million. If you look at those below 65 and have an underlying comorbidity, it's more than 1 billion. About 160 million people work in healthcare or social care. About 1.2 billion are essential workers, and 750 million are adults living in densely populated areas known to have a higher risk of transmission and exposure. So you can see, even if we try to target these on the left side in the gray box, we cannot make it with the 2 billion doses that we are going to think we will have by the end of 2021. So even within this, we now have to further prioritize. We at WHO, we use modelers. We work with a lot of modeling groups to help us think through who are the priority groups, what is the best strategy, that means the optimal use of a vaccine of limited supply for the big, the biggest bang for the biggest buck, but also to address equity and also to address economic and social impacts. How can we keep essential services going? How can we return to normal? What has the biggest economic impact? What is the cost effective? And what is the full value of vaccine? And so I've got a light now in my face. So basically, when we looked at age prioritization, even if the vaccine efficacy is worse in older age groups, if we target older age groups, that's the purple line here, we always have the biggest impact. If we target children or adolescents, we have hardly any impact, even with the same vaccine efficacy. So we developed a roadmap, and this roadmap took into account the epidemiological setting and different degrees of limited vaccine supply. So obviously, the more vaccine supply, the wider your populations. We assumed that all non-pharmaceutical intervention had to continue. We also assumed that vaccine efficacy on transmission, so an indirect effect, would not be known for a while. And also, to be honest, to really receive good vaccine transmission reduction, you also need high vaccine coverage, which we will not achieve initially. So based on this, we developed the 3 scenarios, and I'm only showing you the roadmap for the community transmission, because this scenario applies to most countries globally. So in stage 1, we would focus on health workers and older adults. In stage 2, where we have more than 11%, we would cover all those who have not been covered in stage 1. And remember, even older adults is already above 20%. And we would look at social demographic groups or people who have significantly higher risk because of their professional exposure, because of social demographic factors, etc. We also prioritize teachers because we really want to keep schools going. And in stage 3, basically all the remaining populations should be vaccinated. As for pregnant women, we have some caveats because that will depend on the availability of safety data in this age, in this specific group. So first efficacy results are available now for 5 leading candidates. This also includes the Russian and the Chinese vaccine by Sinopharm. However, they have not really reported the p-values nor the number of events. So we have less accurate data here, but clearly we have very good results for all the other vaccines. And Pfizer is the first one to have obtained emergency use authorization. So let me now focus on the Pfizer vaccine, the mRNA vaccine. They recruited 44,000 persons from several countries. And you can see here that with the primary efficacy endpoint as COVID-19 occurrence 7 days after the second dose, the efficacy was 95%. This is much higher than we had ever hoped for. We had been thinking about 60%, 70%. And so the news of this high efficacy is extremely good news. And even better news is that the efficacy results are same across all the age groups, which is unheard of for many vaccines. So even in those above the 75, we had very good efficacy. It was also similar for across racial groups, and not shown here, but I can tell you that also for comorbidities. So extremely good news. But I want to quickly go into what mRNA vaccines are. So basically, mRNA vaccine have the spike protein encoded in the messenger RNA. The mRNA itself degrades within minutes or hours. And so to keep it stable and for it to really enter the cells, it is combined with a lipid nanoparticle. So it then enters the cytoplasm of the cell, and within the endoplasmic reticulum, it now multiplicates. And then the spike protein is secreted, which then triggers an innate immune response and as well as an adaptive immune response. Of note, this is very important, mRNA does not enter the nucleus. It cannot intercalate with DNA. The mRNA is degraded within hours then by natural occurring mRNAses, by enzymes. So what's only left is the spike protein that is then secreted for the immune response. mRNA vaccines are fantastic, but have major challenges. The challenges is the delivery cost, because it requires an ultra cold chain, which requires capital equipment, a new way of delivering, because you need centers with special equipment for handling ultra cold chains at -70 degrees. It requires extra training, a lot of supervision, a lot of details need to be looked at. So therefore, it has incredible operational complexity that will impact access. It will also delay the access in many countries that do not have all these requirements in place. So this is the last slide. So the good news is the first vaccine to receive emergency use authorization has an incredibly high efficacy. And this efficacy was also seen in older age groups and in persons with comorbidities. Also, all the data so far look like it has a very good safety profile. But the bad news is this very first vaccine that we now have is the most expensive ever vaccine. And it's also the most difficult vaccine to deliver in mankind's history. Never have we had such a most difficult vaccine to handle. So the biggest challenges, in addition to logistics, will also be community acceptance. I think the biggest challenge are conspiracy theories, but of course the practicalities of vaccine supplies, delivery, logistics, and now keeping to equity. Thank you so much for your attention.
Thank you very much. We've been listening to Professor Annelies Wilder-Smith, the first of our two presenters today, and we're going to go straight on to the second presentation from Professor Kang, and then we're going to take questions at the end. So it's my pleasure now to introduce our second speaker, Professor Gangandeep Kang, who is Professor at the Wellcome Trust Research Laboratory and the Division of Gastrointestinal Sciences at Christian Medical College, Vellore, that's CMC Vellore in Tamil Nadu, South India. And Professor Kang conducts interdisciplinary research on enteric infections in child health. Her team has evaluated vaccines in preclinical and clinical phase 1 to 3 studies for rotavirus and also cholera. And she's a member of many WHO advisory committees. She's also the vice chair of the board of Coalition of Epidemic Preparedness Innovations and chairs the Immunization Technical Advisory Group for WHO's Southeast Asia region. So Professor Kang, you're very welcome, and we look forward to hearing your presentation. Thank you.
Thank you very much, Peter, for the introduction, and thanks also to Annelies for an absolutely outstanding presentation that makes life a lot simpler for me, because there's a lot of ground I don't need to cover. So I'm really going to focus on how low and middle income countries should think about COVID-19 vaccines and how we got to the point where we are today. I was actually looking at the ICMDA, and I see that a lot of my colleagues like Anna Pulimuth have given presentations here before, as have my teachers like George Matthew and Vinod Shah. So thank you for doing me the honor of asking me to speak here, and I hope that it will be a useful way of thinking about things. So when we think about vaccines in general, one thing is a given. If there is a new vaccine, it's going to come into high-income countries first, and then it will take years and years and years, sometimes as long as 20 years, before that vaccine becomes generally available to low and middle income countries. Now, that can be a huge problem if there are diseases that don't affect the whole world, but just affect some components of it. So in low and middle income countries, we've lived with infectious diseases for a while that vaccine companies have little or no interest in making vaccines for. And that is really because they don't believe that there is a large enough market for that vaccine at a price that they would be willing to consider. So when Ebola hit West Africa and urban West Africa in about the beginning of 2014, there were actually vaccines available. They had been made 20 years ago, but they had never been taken forward because there was no perceived market for those vaccines. So once Ebola was raging through West Africa, a lot of people began to discuss why this was the case and what could be done to ensure that this did not happen again. So in response to Ebola, there was a new initiative that was established, largely to look at the prevention of outbreaks, particularly for low and middle income countries. And this was the Coalition for Epidemic Preparedness Innovations. The idea here was preparedness. Is it possible to get vaccines ready ahead of time for emerging infectious diseases? And the diseases that were to be considered by CEPI were according to the WHO R&D blueprint, which every year identifies the top 10 threats, but also has on its list disease X, the disease we don't know where it'll come from, when it'll come from, which organism will cause it, and how many people are likely to be affected. The idea was to create a sustainable response to make sure that research moved fast, that vaccines could be developed and used during outbreaks, and this would lead to both access and to equity in terms of responding to outbreaks of infectious diseases. So the CEPI response was essentially around rationalizing and accelerating research for new outbreaks, coordinating resources that were available from industry, academia, governments, philanthropies, and NGOs, and to make sure that there was a prioritization of what the targets were and what platform technologies could potentially be considered to facilitate the development of vaccines for emerging infectious diseases. CEPI was formally established in 2017 and began its funding programs almost immediately. Last year, the first programs for disease X began to be funded. The 5 targets that had been identified by CEPI were Lassa, MERS, Nipah, Rift Valley fever, and chikungunya, all of them from the WHO R&D blueprint. And once those programs had been initiated, a Disease X or platform technology program call was put out, and several awards were made. When the sequence of SARS-CoV-2 was released at the beginning of January, Within 2 weeks, CEPI had already funded 3 programs, including those of Moderna and what is now the AstraZeneca program. Since then, CEPI's funded 12 vaccine candidates, and we heard about the COVAX facility as part of the Access to COVID-19 Tools Accelerator. And CEPI and the Gavi Alliance lead the vaccine pillar of the ACT Accelerator, focusing on research and development of vaccines and the delivery of vaccines, with WHO, of course, taking on the role for prioritization and for building the systems that will allow all of this to happen. So if we think about what should be happening in low and middle income countries, equity and access are really at the heart of everything that we do. Limiting harm is a universal value. So if we think about a vaccine that reduces illness and death, while also making sure that we are mitigating unemployment, poverty, and educational deprivation that disproportionately impact the poor, that is a good basis for deciding how vaccines should be distributed. It's also important to understand that despite the rise of vaccine nationalism, any part of the world is at risk if there are unprotected populations. So this is the basis of the COVAX facility, equity and access, 92 countries that need support, being supported through ODA funding, while other countries also participate in the joint purchase mechanisms as Annelies had described. The other thing that Annelies discussed was prioritization. And in a situation where we are developing vaccines fast, and we know that vaccines are going to be limited, at least initially, it's important to think about the approaches that need to be taken. And the goal here is to protect individuals and health systems and minimize the impact on societies and economies, practically in that order. When we think about differences of location, race, gender, or religion, I think we should make sure that these do not harm or deprioritize disadvantaged population groups. So looking at disadvantaged populations in particular, those who are medically vulnerable and those who have been subjected to socioeconomic deprivation and oppression need to be specially considered when we look within countries at how priorities are framed. We all know of refugee populations where the local population is protected first, and those who are considered as foreign are not allowed access to many preventive and healthcare services. Now, I'd like to move a little bit to India and just talk about the scenario using India as an example. India actually makes more vaccine than any other country in the world. It has major vaccine manufacturers that have been making vaccines for supply to UNICEF for many years now. And they make WHO pre-qualified vaccines so that you have vaccines of assured quality for immunization programs around the world. These vaccines tend to be high volume but low value vaccines because they are made based on older technologies. So many of the vaccines that we have cost cents a dose. And it is one of the reasons why Gavi has been able to really stretch the funding that it has received to immunize children around the world. Now, if we look at what's happening with COVID-19, Indian vaccine companies used to be old technology companies. We are in a situation today where we have multiple different vaccine platforms, and there is a remarkable change in how companies in India have approached the making of vaccines against a new pathogen. So we already know about the major players. We know about the biotechs that have now come into making vaccines. Moderna, Inovio, Clover are examples of those. But if we look at all of the platforms shown here in red are Indian companies that are making vaccines based on the platforms shown here. Zydus Cadila is making a DNA vaccine. There are inactivated, there's a self-amplifying RNA vaccine being made by Genova. There are live attenuated vaccines, replicating and non-replicating vectors, and subunit and VLP vaccines being made as well. So there are a lot of different candidates. Some of the candidates there are 5 candidates that have been in or through phase 1 and 2 studies, and there is one candidate that is in clinical efficacy studies, which is an inactivated vaccine from Bharat Biotech. The other candidates that are being made in partnership with other companies, the Gamaleya and the AstraZeneca vaccines, are being produced by the Serum Institute of India and by a company in India called Hetero Pharma, which will be taking on the responsibility for making 100 million doses of the Gamaleya vaccine. So last week, we had pretty striking news in India where we had Pfizer applying for emergency approval to the regulator. We also had the University of Oxford, AstraZeneca, the Serum Institute manufactured product, and Bharat Biotech, which has its inactivated vaccine currently in phase 3 trials, applying for emergency approval. Pfizer has no data in Indian subjects. No vaccine has been approved so far in India without such data. But DCGI, our regulator, the Drug Controller General of India, can decide to waive clinical trials. Serum Institute is manufacturing vaccine and is a major supplier to the COVAX facility, but the clinical trials in India are relatively limited. They have 1,600 subjects that have been recruited and given 2 doses. Immunogenicity data from these subjects is awaited, and they can have, based on the immunogenicity, a bridging strategy that will give us to the phase 3 data from UK and Brazil's AstraZeneca trials that were published in Lancet 4 days ago, I think. Now, the interesting thing about the Serum Institute study is that they've already applied without the immunogenicity data. And at least initially, there was a lot of discussion about which data they were going to bridge to. I think everyone is aware that there were 2 different combinations of vaccines that were given in the UK and in Brazil, with a low dose and a standard dose, or 2 doses with the standard dose. And there was a much higher efficacy seen in the small number of participants that were in the low dose, standard dose combination. So when you have unclear communications about why you have a difference in results and unclear communications about why you wound up with different combinations of vaccines, it creates a lot of doubt in people's minds. And this is amplified, if anything, in LMICs like India. We'll come to that in a minute. Now, Bharat Biotech has completed its phase 1 and phase 2 studies, and it has initiated its phase 3 studies. Currently, it has about 8,000 participants that have received their first dose. Now, all three companies have applied for emergency approval. And this creates a perception that the companies are trying to take a shortcut and not follow due process. So when you think about India as a vaccine manufacturer for the world, it's worrying if this is allowed to go through. Fortunately for us, the Drug Controller General of India has so far at least decided that it is not going to allow the Serum Institute and Bharat Biotech products to move forward without additional data. So this is something that we need to wait and watch. Now, even when we do have a vaccine, what is the preparation that allows us to distribute that vaccine? So in India, our Health Secretary, our Ministry of Health and Family Welfare have been following pretty much along the guidelines set by WHO, where 30 million healthcare and frontline workers will be immunized first. There are 10 million doctors, MBBS students, paramedics, etc., and 20 million of what are considered frontline workers. And these will be followed by 260 million individuals who are above 50 years of age or have comorbidities or require special care. And this will include not only those who are in public healthcare facilities, but in private healthcare as well. According to the government, there's sufficient supply of vaccines and everything else, and all of this will be done over a period of 7 months from January to July. The government keeps stating detailed plans quite regularly, the tiered system for identifying populations and delivering vaccines, and most recently, the government has developed an app that will allow people to register for getting the vaccine so that people can be communicated with directly. Now, the Duke Global Health Innovation Center has reported that India has an advanced market commitment of 1,600 million doses. This is not information that is widely available in India, but according to Duke, this commitment has been made with 3 leading manufacturers, including the Serum Institute of India, which makes the AstraZeneca vaccine. So when a vaccine is approved, how will it be delivered in India? The government intends to purchase it all. We already have the largest immunization program in the world with 9,000 cold chain points, 28,000 locations, and the intention is to use agricultural private capacity for additional cold storage. We have no -70 within the immunization system. We have limited -20 that was there for the polio immunization program. I mentioned the repurposing of what was called our electronic vaccine intelligence network to CoWIN, which is now an app that will be available not only to healthcare staff, but to the general public for booking vaccine doses. And it's intended that we will have a digital identifier for identification of individuals who are registering for these vaccines. Now, this, of course, is a huge challenge in a country where documentation has always been extremely challenging. There is a tiered system for information, for distribution, and this is for populations of 5,000, 30,000, and up to 1 million. And committees have been established for preparedness and steering. So if we look overall, we are seeking to accelerate the entire process of developing vaccines and delivering vaccines. Is this going to be challenging? Yes, of course it is. And if just to end, these are the questions I get asked most often. Are the vaccines safe? Have we cut corners? Who gets the vaccine first or why? Which are the right vaccines? How do you do head-to-head comparisons? Can we actually deliver some of these vaccines? Who's going to pay for them? We can't afford a $20 vaccine. And the AstraZeneca vaccine is intended to be priced at about $3 a dose for the Indian government. But that is considerably more expensive than the 60 cents a dose that we pay for our current most expensive vaccine. So I'll end there and thank you for your attention.
Well, thank you very much, Professor Kang. I'm Dr. Peter Saunders, the CEO of the ICMDA, and we're now moving into a time of questions. So we've heard from two excellent, eminent speakers, a huge amount of information, very comprehensive, and the questions are rolling in. So I wonder, Professor Kang, if we could just start perhaps with some of the questions that you've raised, which are most common. And I think you've certainly characterized them here, but the big question for developing countries, who is going to pay for the vaccine, especially when those that seem to be most effective are also the most difficult to deliver? Yeah, who's going to pay?
I think this is going to be a challenge for all countries. While we can be reasonably sure that the COVAX facility will do the best it can to get doses to at least 20% of the world's population, we do have to think about the other 80. We've never had a situation before where the entire world has needed to be vaccinated. In many low and middle income countries, we've never had an adult immunization program other than tetanus for pregnant women. So for governments to start thinking through how they are going to find the resources for the remaining 80%, that's going to be important. I know that the World Bank and the regional banks like the Asian Development Bank are speaking with countries to try and figure out what their needs are and see whether it is possible to make loans to the countries in addition to the grant of vaccines in order to be able to buy enough doses and deliver them.
Do you think vaccines can end this pandemic on the basis of the evidence that we have already?
I think vaccines will certainly accelerate the process for control. Will we be able to wipe this virus out? Personally, I think that's a little unlikely, given that we now have evidence not only of a large amount of asymptomatic infection, but also of the ability to cross over back into animals and into humans. So eradication will be a challenge, but very good control is feasible with vaccines.
Thank you. Professor Wilder-Smith, I wonder if we could come back to you on this whole question of herd immunity, because there is a lot of misinformation, I think, going round. Some people are claiming that we have herd immunity already because of the level of unmeasured cell-mediated immunity, for example. What do you think is the level of immunity in the population at the moment, generally, and what percentage of the population would need to be vaccinated in order for us all to be safe, do you think?
Yeah, good question. So there's a seroprevalence tracker. And so we do have a feel about seroprevalence as it's evolving around the world. And it differs highly, you know, but I think in general terms, the average is about 10%. Now, the herd immunity can be calculated if you know the R0, the reproduction number. Now, we know that the reproduction number without mitigation measures is between 2.5 and 3. And the herd immunity, in brief, is 1 minus 1 divided by R0. So anyway, you need about 60%. 60% to get herd immunity so that your R0 is clearly driven below 1. That doesn't mean you have no more cases, but it's not a public health problem anymore. So to achieve a 60% coverage immunity, either through natural immunity combined with vaccine immunity, will take a long time and will cause many deaths. Therefore, as I agree with Professor Kang that the initial, that the virus will be around for a long time, but the fear and the anxiety and the problems are the deaths. So if we can at least cover and protect the high-risk populations, I think then the threat of this pandemic is reduced. So I think that's the first aim. And the second aim is then really also to reduce transmission in general. But I also agree with Professor Kang, because of the zoonotic spillover, we will likely have to face this virus for a long, long time.
Okay. A very popular question here. Why are we still recommending vaccinating patients who've already had COVID? If we don't think there's lasting immunity from natural infection, why do we think a vaccine is going to help us? Many people worry about these things.
Yes. So we currently would not exclude someone who has a history of recent infection from vaccination. We definitely will not do any screening before we do vaccination rollout. Obviously, the benefit initially will be higher in those who have not had a prior infection. But we do know that a small minority have reinfections, that natural immunity may not be lifelong, although we now have evidence of good protection for up to 6 months. We would at least not exclude those who had prior infection.
Perhaps a tricky question, but which vaccine would you put most faith in? And I realize this is very complex because what we know there's variability in cost, logistics and delivery, cold storage and so on. But it seems a lot of lower middle income countries are also opting for the Chinese or the Russian vaccine, seemingly because it's more easily accessible. So would you like, you've spoken very highly about the Pfizer vaccine, but would you like to comment briefly on the others and whether, especially whether people should be worried about vaccines from Russia or China?
Sure, do you want to go first?
Sure. So I think WHO set up a target product profile, and it looks like that the vaccines that are based on the mRNA platform and the viral vector platform have both exceeded what was required from the target product profile in terms of efficacy. When we look at vaccine trials, the vaccine trial design is not identical. So if you see a vaccine that has 90% efficacy and another one that has 80% efficacy, it does not necessarily mean that those two vaccines are very, very different. We will know that once we do impact assessment studies in real life. So I think what we should go with is really how useful is this vaccine for my country's program? How usable is it? If it is a vaccine that fits into existing cold chain capacity, is a less expensive vaccine, I have reasonable data on safety and efficacy from this vaccine, it's been approved by a regulator using a data set that I think is sufficient, I would advise my country or any other country to go for that vaccine. The question really is what constitutes an adequate data set for regulatory review. And at least in terms of the initial approval of the Gamaleya vaccine based on 39 immunized individuals, that was insufficient. If you look at the Chinese data as well, we don't know what data were used to inform decision-making. As long as there is clarity on what data were used and that those data were sufficient to demonstrate with a reasonable degree of confidence that these vaccines are safe and efficacious, we can use them. We just don't have clarity on that for the Chinese and the Russian regulators. We do for the MHRA and the FDA.
Thank you. Annalise, do you want to add anything to that?
I think she spoke perfectly.
It's
That's very
pretty clear, the clearest explanation I've ever heard. There's a lot of anxiety about mRNA techniques just because they're new. And shouldn't we be using old technology that's proven, people are saying. But let me just read out a few of the sorts of questions we're getting. With the new mRNA vaccines, what happens to the RNA? Does it go into all cells or just some? Will it mean that those cells go on indefinitely reproducing the spike protein? And if is there any danger that human cells will display the spike proteins and therefore be the subject of immunological attack as well? So could you just talk around those sorts of anxieties for something that's very new?
So may I take that?
Yes, Professor Kang, please.
Okay. So I think one of the things that we need to remember is that RNA is actually a very delicate molecule. It breaks down very easily. And one of the major challenges that we had in making mRNA vaccines was that we were unable to protect the mRNA enough for it to get inside of cells. Now, because we have these little lipid nanoparticles, little, essentially fat protecting the RNA and allowing the lipid nanoparticle to fuse with the cholesterol in the cell membrane, we have a delivery mechanism. So you give an injection and the RNA, it can get inside cells that are in and around the muscle. There is a limited amount of RNA that is given. Now, RNA cannot replicate by itself. The RNA is encoding the spike protein. It delivers its message and it breaks down. There is no RNA left. So you're making spike protein in one cycle of protein production. And that is then released from the cell and you respond to it. So even though we use the term factory, it's a one-off factory. The human host cell is a one-off manufacturing mechanism. It does not stick around. And because, again, RNA is only in the cytoplasm, it doesn't go to the nucleus. So there is no integration of the RNA with the host nuclear material. Also, people talk about RNA sticking around and making genetically modified humans. That doesn't happen because we don't have a reverse transcriptase that could convert the RNA into DNA and then allow it to integrate with the host chromosome. So RNA is really a very, very safe way of delivering a signal on how to make spike proteins.
I totally agree with Cherry, and I do actually think that it will revolutionize also future vaccines. It's a very rapid development, as we have seen now. It is very safe. It's not a live attenuated vaccine, doesn't have a disadvantage of a live attenuated vaccine. It's almost like an inactivated vaccine. It does have, we know already now, it does have quite some reactogenicity. That means quite some local pain, headache, fatigue, but that all abates after about 48 hours or 3 days. So people need to be informed. Yes, it is painful, but no, so far we have not seen any long-term safety concerns. Obviously, I agree. We do not have a long-term observation time. Our median time now is 2+ months, but the safety data that we've seen look very encouraging.
Just again on the mRNA vaccines, you've talked a lot already about their need for cold storage, the logistics and difficulty, the tendency to degenerate, and the high cost relative to AstraZeneca. Is this going to be a problem, do you think, in developing countries? Professor Kang, you've already talked quite a bit about India, but is this going to preclude its wide use in the places where it's very greatly needed? Annalise.
It's going to be really hard to deliver. So even if through the COVAX facility, it was possible to make an allocation of a small number of doses, that would depend on the country readiness to distribute those doses. And we know that very few countries have the capacity to now put in. No one has -70 capacity. You would have to establish that. And that is an additional investment. And Gavi is not intending to provide grants for ultra cold storage.
Okay, a lot of questions about the role of pharmaceutical companies and their vested interests in this whole thing. We know a lot of people don't trust big pharma, and let me just give two of those questions. The early vaccine data looks too good to be true. Do you think we can trust it, and are you confident that the private companies are being transparent with their results? That's the first question. And related to that, what do you think about the UK government decision to allow indemnity from legal action to Pfizer over their COVID vaccine? Is this going to undermine confidence? So perhaps to talk about the reliability of the data and any vested interests.
The data that we have seen the data, and the data are the data that we've seen. I think what underpins the reliability of the Pfizer vaccine is that Moderna has about the same results. High efficacy, good safety, both are more painful than the second dose. They all seem to mirror each other. So I do believe those data. Yes, we lack long-term data. We will have to live with this. This is about a benefit-risk assessment. The benefit clearly outweighs the risk in this current moment, and the pandemic is causing too many deaths. Indemnification. Now, I am not a legal person, but this is what I understand. So every vaccine, after it's introduced, there is a risk transfer solution. This is a legal process that takes time. We don't have this time. So in this case, all countries must, and that is for all countries, not the UK, all countries must indemnify manufacturers, donors, and distribution against any losses they incur. On the other hand, we have to give the individuals the right, so the individual who has received the vaccine, the right, if they do suffer a serious adverse event, that they will have access to mechanisms to provide compensation. That's all I can say, and the indemnification contracts are still, in part, still being negotiated. It's part of COVAX, and it's part of the full package of being a COVAX recipient of vaccines.
Thank you. We're getting questions about the effectiveness or the dangers to immunocompromised individuals, particularly perhaps people with HIV or who are immunocompromised for other reasons. Are they going to be able to be effectively vaccinated, or are there any safety concerns?
So, okay. So the AstraZeneca vaccine is actually being tried in a population that is both HIV exposed and HIV infected. So we will have safety data from that. In terms of immunosuppression, in the early phase trials, people who were immunosuppressed were excluded from the trials, and a lot depends on what immunosuppressant you are on for what period of time. So I know that there is a lot of concern for people who have immunosuppression for a variety of reasons, including transplants and those who are on treatment for cancer. I think these data will emerge over time, and there are plans to do smaller studies with certain kinds of vaccine products that we think will be safer than others. So looking at non-replicating products, essentially.
Thank you. Annelies, if I might come back to your presentation, you talked about the degree of uncertainty that we have over a lot of the science simply because the trials haven't been run for long enough. So questions like, will it prevent transmission or just infection? And how long will the immunity last? I heard from someone in North Africa yesterday that the Chinese vaccine, they're being told, will only last for 1 to 3 months. I'd never heard that before and didn't know how they could have the data for it. But could you comment on those questions that we really just don't know the answers to yet, and what effect that might have?
So those questions require post-authorization vaccine effectiveness studies, both to determine the duration of protection and then the timing and need of a booster. Second, also for long-term safety, but also, the waning of efficacy, etc. So we need to be reminded this vaccine is currently not fully licensed. It's only under emergency use authorization. So the company has an obligation to deliver and to provide longer-term data. The ethical dilemma that we have is whether we can keep the placebo group. And there's a lot of discussions. We only have a few minutes left, too much for this, but I can tell you we really, scientifically, we would need to keep the placebo group to look at all these questions. I hope that answered it enough for the time being.
Thank you very much. Professor Kang, do you want to add to that at all?
No, I think that's fine. Yeah, it's one of those where once you start talking about detail, it just goes on way too long.
Yeah, so. Okay. Thank you. And this next question, several of the viral vector vaccines, AstraZeneca, Janssen, and others are produced using the human cell lines HEK293 and PER-C6, which were derived from cell populations which initially came some decades ago from aborted fetuses. I think one in the Netherlands in the 1970s gave HEK293. I know there's some anxiety in the Christian community about this. Any comments on general principles here? I know it's a little bit outside the breadth of your talk, and you may not want to comment on it, but.
Yeah, maybe Peter, you're the better expert, but what I can say that these original cell lines were used for the production of various vaccines that we are using in the moment, even varicella, hepatitis E, A, etc. So, but the vaccines themselves do not contain any fetal material itself. So, and for the mRNA vaccine, there's nothing in it. It is a total new platform. But for the adenoviral vectors, as you said, it does have the original cell lines. I do know that the Catholic Church years ago made a statement and said that the population benefit would outweigh those concerns. I lived in Singapore, where we had to give vaccines, meningococcal vaccines, to Hajj pilgrims, and the Muslims communities were very, very concerned about the porcine element of some, that it would be non-halal. And also the Muslim communities or experts said the public health benefit outweigh here those ethical concerns.
Yes, thank you. Thank you very much. I could refer you to, there are 2 good articles on the ICMDA blog that deal with this question by Professor John Wyatt and by John Ling, respectively. And they interestingly both take the view that because even though obviously we condemn abortion and we condemn the whole idea that the end justifies the means, but when you're looking at cell lines that were derived 4 or 5 decades ago, where there's no complicity or involvement or intention involved, that it's a different kind of question. And as you say, there's no aborted fetal matter in any of these vaccines, and they've been used very, very widely in a huge amount of research and in producing many other vaccines that we use regularly. Well, I think sadly we've run out of time. I'm sure we could go much longer. So thank you once again, both of you, for your talks today and for answering the questions so ably. And thank you everybody for attending today and hearing these presentations. Do tell people about ICMDA webinars, and we hope to see you again soon. God bless you. Thank you.